Now, if we examine the region of the gene, the DNA, right? What happens is, you see that, hey, DNA itself is double-stranded, right? It has two strands. So, if you look at this, you can imagine that there's a complementary strand of DNA to this gene, promoter, transcribe region, and terminator. There's actually a complementary strand, okay? Like that. You see that? Okay? So, if I were to decode this region, this corresponding region of the complementary DNA strand, what would be the sequence of this transcribed region? A. So what would be the sequence here? What would be the base here? T. T will be A. Now remember, this is the complementary DNA strand, which is why this is T, right? Don't forget, DNA comes in double strands. Okay? So this will be C, G, T, A, C, G. Okay? Now, I want you to notice something. Then we can understand why we use the words in the way it is used. You look at this sequence here that is complementary to the transcribed region. Okay? Notice the sequence of bases. Then I want you to pay attention to the sequence of bases on the RNA transcript as well. What do you see? Very, very similar, isn't it? The only difference is instead of a T, it's a U. Right? Okay? So the sequence is, it's almost identical except for U in place of T. Okay? So you see that the transcript has a very similar sequence to the sequence of DNA that is opposite the transcribed region. That's because of Chargaff's rule. It must be this way, isn't it? Okay? Alright, so how does that tie in with the words that we are trying to learn now? So, now remember, what is the job of the RNA transcript? It will leave the nucleus of the cell, go into the cytoplasm, and then the other organelles, specifically the ribosome, will then use the code on the RNA transcript, on the mRNA, to make protein. Right? So in other words, the ribosome is the one that makes sense of the message, isn't it? Tries to use the message, the code, to make proteins. Okay? So the sequence on the mRNA or the RNA transcript is the sequence that makes sense to the rest of the cell. You get that? Okay? Which then causes the, or I wouldn't use the word cause, but which then we can apply the sequence here. So this strand or this sequence that is complementary to the transcribed region is the one that makes sense. You see that? Because it is very similar to the one on the mRNA. Okay? So which is why we call this sequence that is opposite the transcribed region, this sequence of the DNA, we call it the sense strand, the one that will make sense to the ribosome. That's how it is named. Okay? So what it also means is the transcribed region, because it is complementary or opposite of the sense strand. Okay? Then this region here, we call it the anti-sense strand. That's how the names came about.